In this video, we will discuss about how incretin is secreted and how incretin works on the beta cell. In the first time, how the incretin is secreted by the wall of the GI tract. When we eat sandwich, this sandwich is digested, and in our small intestine, a lot of glucose will be present. And this glucose and fatty acid triggers the release of incretin by enteroendocrine cells, which is located in the duodenum predominantly. And also the glucose and fatty acid induces the release of incretin, GLP-1, the glucagon-like peptide 1, by enteroendocrine cell, which is located in the small intestine and ascending colon. And this GLP-1 and GIP are all incretins. And how this incretin works on the beta cell to increase the production and release of the insulin. Let me move our panel to the right side. Because of the space, I copied the node incretin here. So this copy is copied node of incretin because of the problem of space. And this incretin moves through the blood vessels and reaches to the beta cell and activates GLP-1 receptor, the glucagon-like peptide 1 receptor, and also the exenatide, which is the GLP-1 agonist, GLP-1 receptor agonist, or the GLP-1 receptor agonist as GLP-1 analog. This is actually used as a drug for diabetes mellitus. If the GLP-1 receptor is activated, the GS-alpha molecule is produced. This is small G protein, and this activates the enzyme called acetyl cyclase. And this enzyme converts the ATP to cAMP. And this cAMP activates two signaling molecules called EPEC and pKa. And this pKa activates the IP3. This IP3 induces movement of calcium from endoplasmic reticulum to the cytosol. cAMP also activates the EPEC, which activates the RYR, the ryanodine receptor, on the ER. So this RYR also induces the movement of calcium inside the endoplasmic reticulum to the cytosol. So cytosolic calcium level is increased, which induces the secretion of the insulin. And at the same time, the pKa activates PrEP, which is one of the transcription factor in the beta cell. The PrEP increases the production of insulin inside the beta cell, which further enhances the secretion of the insulin from the beta cell. And also the So I know the one more mechanism that PKA directly phosphorylates the voltage-gated calcium channel. So this link here, this link. Let me see more clearly. This link. So PKA activates the opening of the voltage-gated calcium channel. So causes this causes the movement of extracellular calcium to the cytosolic calcium. So this further increases the secretion of insulin from beta cell. And let me see the other mechanism here. Yes, from here, let me explain the other mechanism. Further increases the production of insulin from beta cell. This crab increases the transcription of IRS2, that is insulin response segment 2, and this acts on the IGF-1 receptor to activate the PI3K. IGF-1 receptor binds with IGF-1 or insulin. This insulin is the same insulin as here. So the produced insulin acts on the beta cell itself, so the autocrine action of insulin. And if this IRS2 level is increased in the beta cell, the IGF-1 receptor are more actively activates the PI3K. And this PI3K activates the AKT or PKB, which inhibits the FOXO1. And originally, the FOXO1 is inhibiting the PDX1, which is the powerful proliferating agents for beta cell. So the inhibition is activation. So AKT activates PDX1, which further increases the production of insulin by proliferating the beta cells. And AKT also inhibits the apoptosis of the beta cell, so the death of the beta cell, which further increases the number of beta cells. And also the other mechanism is present. The GLP-1 receptor activated by incretin. This incretin is the same incretin produced by here. So the GLP-1 or GIP released by enteroendocrine L cell or enteroendocrine K cell. This incretin moves via the blood vessel to reach here, or the exenatide, which is a drop, activates GLP-1. We already discussed about this pathway, GS-alpha acetylcyclase, CAMP, EPEC or PKA, which activates RYR or IP3, which increases the movement of the calcium from endoplasmic reticulum or extracellular space, so the increased calcium concentration inside the beta cell, which increases the secretion of insulin by the beta cell. And let me discuss about the other pathway here. So GLP activation of GLP-1 receptor also activates the CSRC, which is the little tyrosine kinase nearby the cell membrane, which activates the ADAM, which is a kind of integrin molecule present in the plasma membrane of the beta cell, which also transfers signal to the BTC, beta cellulin, which activates finally the EGFR. So this EGFR, when this EGFR takes signal from this cascade GLP-1 receptor, CSRC, ADAM, and BTC, the EGFR activates the small G protein called GAP1 and GRB2. And this small G protein activates PI3K. Further activates PI3K. And this PI3K again activates AKT, which increases the production of insulin and proliferation of beta cell and inhibit the apoptosis of the beta cell. And this is all the mechanism of the how the incretin is produced and how the incretin works in the beta cell. Thank you for listening.